This is Deepak Chopra, and we are continuing our conversations with luminaries, influencers, cutting edge creative thinkers, and just about anyone who can help alleviate the immense suffering that's happening in the world today because of unconscious behaviors and the link between climate change and social injustice, economic injustice, all kinds of problems, health, well-being are being seen now when we look at well-being in a holistic manner. So we have a very distinguished guest today, Britt Koskella, Britt Koskella, an eminent scientist. And first of all, thank you, Dr. Britt, for joining us. Um, the way we do these conversations is I like to ask our guests to tell us a little bit about their life, going all the way back to where they grew up, their family, their education, their influences, and how come we are having this conversation. So take it from there.
Wonderful. Thank you. I'm very glad to be here, and it's wonderful to meet you. Um, Goodness, what highlights my development of how I got here? I will say I was not a born scientist. You know, some people say that I knew I was going to be a biologist from the ripe old age of 1, and that was not my path in any way, shape, or form. I had a fairly normal childhood, if you will. We moved a lot. My dad was in the military in the Air Force, and so I,
Mine too, brother.
yeah, and so I got good at reinventing myself. Every move, I became a slightly different version of myself. And that was very freeing and allowed me to try out many different paths and many different types of myself, different versions of myself. And when I went to undergraduate, I was at the University of Virginia, and I was started as a psychology major. I had no idea what I wanted to do when I grew up, whatever that even meant. And I got quite interested in how I learned about evolution, evolutionary biology, and I loved the idea that everything that was quirky and unusual about me and those that I met were somehow the relics of this evolutionary history, and the idea that everything is a trade-off. And so if I'm good at one thing, I'm less good at another, because that's what evolution and natural selection does, is it selects for optimal fitness in a given environment, but that is not perfect. There's nothing perfection about it. And it's actually all about imperfections and balancing the imperfections and making imperfections strengths. And so I absolutely loved learning about evolutionary biology, and it completely changed the way that I think about myself as well as the world around me. Uh, but I knew that thinking about human evolution was not for me. And so I got a job as a research technician. And what I did for my job was an experiment that we call experimental evolution. The idea that you can take, in this case, it was a fungal pathogen of a plant, where we decided what selection pressure we wanted to put onto this fungal pathogen population. And in this case, we wanted to understand how it adapted to its host, this plant in the field. And so we put the pathogen where we wanted it. We controlled the selection, and we changed the outcome of evolution. We changed the evolutionary trajectory of this pathogen. And seeing that occur in real time, and seeing that evolution isn't something that just happened over very long timescales, but it happens every day, really, again, was a frame shift for me. It completely changed how I think about what we as humans are doing to the world every moment. And of course, the most famous example is antibiotic resistance. You know, when we take an antibiotic, we are imposing selection. We are changing the evolutionary trajectory of bacteria in our gut. And so I was really hook, line, and sinker. I loved the idea that you could test these ideas and see evolution happen. I loved it as a teaching tool as well. It's a wonderful way of engaging students' minds. But I also knew that that's what I wanted to do with my life. And so I did my PhD at Indiana University, where I worked on snails and trematode parasites that infected them. And they're asking these questions about the Red Queen hypothesis of how parasites maintain biodiversity, which we can talk about if you're interested later. And I loved again this idea that these interactions among species change diversity on Earth. But I wanted to do something a bit more applied. And so when I got my faculty position here at the University of California, Berkeley, I decided to take this love of evolutionary biology and this love of understanding ecological species interactions and apply them to agricultural settings. And so that really got us here is thinking about how everything that we do as humans on Earth is changing how species interact, how evolution happens, and both understanding the negative consequences of that, but also thinking about how we can leverage it to make a better world.
Well, that's very comprehensive and very fascinating and very interesting. I want to come back to evolutionary theory, including Darwin, but not right now. We can go into it in a few minutes. But right now, I think I'd like to ask Rajnish um, what his fascination is with your collaboration with him on this very interesting technology. So go ahead, Rajnish.
Oh, and don't forget to unmute.
Yes, thank you, Deepak. Um, I met Britt, I think almost 3 years ago. I went to a seminar that she gave at UC Berkeley, and I was very interested in what she was doing. And at that time, I was also starting a company called i-Cultiver, where I wanted to invite people like her who are doing really cutting edge academic research to see how we can bring the academic research out into application. And so since then, we've been working together actually on a community of bacteria, which we call Phyllosphere. That's why I have that name up there. And we've been studying together these bacteria on in greenhouse grown plants and on field grown plants with amazing results. And I think Britt will share some of those perhaps during this conversation. So that we've been working together, and I've just truly enjoyed learning and working with Britt, and look forward to working more together.
So, may I call you Dr. Koskella or Dr. Britt?
You can call me Britt, absolutely. Whichever, yes.
So, given what Rajni shared, these bacteria, phylo, whatever you're calling them, these are bacteria that change the ecosystem, I'm guessing, of the relationship of genetic information in the soil. Would that be an appropriate way of characterizing what you're doing?
Almost perfectly until the very end. And that's actually
But
what's
let's
the
go
most
back. Let's
exciting.
go back to what you said. Almost very good.
Almost perfect until the last word, which was the soil.
Oh,
So what sets what we're doing apart is we do very much care about what's in the soil, and you're absolutely right that the soil microbial communities are critical. They're critically important to plant health, human health, and they are where most people study microbes and plant microbial interactions. And what my lab does, which is a little bit different, and it's not just me, but there's a handful of us working in this space, is looking above ground at the what we call the phyllosphere, the leaves, the flowers, the fruit. And so the phyllosphere above ground is often an afterthought in terms of the microbial community, but it turns out that the microbial community there is incredibly important to plant function, growth, yield, and nutrition of the fruits and vegetables produced. And so these are microbes that live above ground on leaves.
so are we then, in a way, metaphorically, again, excuse my ignorance, I'm not a scientist, but I'm interested in science. I'm a physician by training. What are you then, in a way, talking about what we would call the microbiome of the plant or of the,
Yes,
okay.
absolutely. And going, since you mentioned being a physician, I do think that there's real parallels with the human microbiome. And I often think about the root microbiome below ground as being equivalent to our gut microbiome, for example. That's the powerhouse where we need microbes to turn nutrients around and make them accessible to the plant. And I often think about the above ground Phyllosphere microbiome as the equivalent of our skin microbial communities, our skin microbiome, which is also incredibly critical to human health and acts as the same interface between the host organism and the climate, the air, the wind, bugs that land. So I that's how I like to think about it, is the sort of protective layer above ground where the
I think,
plant interfaces with the world.
Poonacha, one second.
Yeah.
Britt, then just share with us what are the clinical or other implications of this for the ecology of the planet, for climate change, for agriculture, and for our health and well-being. Then I'd like Poonacha to come.
Yeah. So I'll answer on two different levels. So, in practically speaking, the product itself and the work that we're doing in this agricultural space is to try to understand how agricultural practice has changed the natural microbiome of the plant. And the answer is it has, in very impactful ways. And that's not surprising because so much of agricultural history, and this is very similar with clinical history, is that we've thought of our task as managing microbes, as reducing bacterial densities, because typically we've thought about pathogens, bacteria as pathogenic and bad. And so much of what we do in agriculture or clinical health is reducing germs, right? Reducing bacteria. And of course, now we're realizing what a mistake that was, and how these microbiomes, whether they be human microbiomes or plant microbiomes, are a fundamental part of that organism. And so by everything we've done to reduce the bacterial burden or the fungal burden has had these negative consequences. And so this product in particular is trying to redress that problem, that reduction of diversity. But the second level I wanted to answer your question on is a more philosophical one, I suppose, which is my lab, what we think about, we work at this practical level of developing products and testing bacteria, but we also work at this very theoretical level of understanding the drivers of diversity and how you get a healthy, diverse microbiome. And so I think that the questions that we're asking on a plant were the mechanisms by which we can increase diversity of a microbial community or increase diversity of a plant community on Earth. These are all very similar processes, and the same thing is going to be true for the human microbiome. And so I suppose I like to think that we work on plants both because they're important, but because they're really nice model systems for asking these more general, big picture, blue sky questions as well.
Poonacha, since you are spearheading this whole rewilding cities, Lake Nona and other communities, what do you think are the practical applications for some of the efforts at the Chopra Foundation?
I think, first of all, I'm really excited about this. I've been spending some time really looking at your body of research, Dr. Koskella. So I really like the way you distinguish the rhizosphere and the phyllosphere, right? What I was also fascinated was that the rhizosphere has 117,000 studies,
Yeah.
and you had the phyllosphere only has 15,000. So but you also say that the biomass, 60% of the biomass is the phyllosphere. And you can actually measure and manipulate the phyllosphere more effectively. So I find that a huge, why are we missing this huge opportunity for us to really make a difference? So I look in the Chopra Foundation, yes, we can go agricultural science, we can do a lot, but we're today living in cities, right? And we have community gardens. Now in Orlando and Lake Nona, the mayor is also looking at making edible gardens. So you can actually change your front yard into edible gardens. So I look at this as a huge opportunity for us to look at looking at microbiome diversity in cities and urban areas, and really bringing health and wellness back to our children, to our communities. We could, like childhood obesity, right? Some of those things, I think it starts with food. So from a Chopra Foundation, I would love for you to kind of share your insights into the future of cities, how we could leverage the work you're doing with microbiome diversity, And maybe walk us through the lens, which I love what you've done, is looking at environment, resource, the parasite and predators, and then the host. How does that model help cities design better communities?
So if you'll allow me, I'm gonna answer the question by taking you on a slightly wild ride. So I'm gonna talk about the development of infant formula, and hopefully it will become very clear why I'm doing that in a moment. When when it became clear that we needed infant formula because infants were starving. Of course, how we started to do that is to put food together that we understood that people need to survive. So we created a formula that was things that were digestible. And if you watch the trajectory of the evolution of, if you will, of that product, infant formula, it has become so complex now, right? Things that we would have never dreamed of putting into infant formula at the start, we're now creating this. And we're still not perfect. There's still a big difference between the nutritional value of breast milk and infant formula, but we're getting a lot better. And so one of the most recent pieces of the puzzle is that it turns out that breast milk has these oligosaccharides that are not digestible by babies. And so they were ignored by the health community for a long time because why are they there? They're not nutritious. We're not going to put them in infant formula. Well, it turns out they're there to feed the microbes. They are there to cultivate the infant gut microbiome. And when that final piece of the puzzle falls, then everything starts to make sense. And now we're in a position where we could, in theory, make an infant formula that would come as close as we've ever been able to mimicking all of the holistic health benefits of breast milk. Okay, so the reason I tell this story is because I see a lot of what we're doing in urban ecology and urban planning gardens as very similar, right? We have to start somewhere. We know that we're starting far from optimal. We're starting with soil that is not as diverse as a soil in a forest. It has reduced diversity. It probably also has some pathogens in there that we don't want. And it doesn't have the nutritional value of soil from a diverse field, for example. So how do we, I think the plan is to slowly start creating these mini microcosms that come as close as we can to nature. And one way you should think about doing that is there's this idea that, using the idea that diversity begets diversity. So, and I mean that in saying, if you want to cultivate a diverse microbial community, for example, then you need a diverse plant community. If you want to cultivate a diverse plant community, you need a diverse microbial community, right? And of course, that includes insects and pollinators. And so I feel like what we've been doing inadvertently for so long now, and this is especially true in city planning, is reducing diversity. We plant the same tree all the way down the streets, and this is going to have and has clear knock-on consequences to microbial diversity, insect diversity, and so forth. And I feel like there's very good evidence that essentially leads to a vicious cycle of reduction in diversity. And so I see what we're trying to do now is just rebuild diversity from the ground up, literally from the microbes up.
Very good. Rajnish, with this Phylostart, what are your aspirations? What do you think Phylostart will ultimately help in accomplishing?
Mute.
Sorry. What we have tested is the use of Phylostart early on in plant development. That's why the phyllo from Phyllosphere and start when the plant is starting, when it's very young, about 4 or 5 leaves, and we can spray the Phylostart on the leaves. And we've actually found that even when the plants that are grown in the greenhouse, they transfer to field, those bacteria, some of them we are able to track, and they are there. So it colonizes the leaves. And so the aspiration is that what we have discovered here is that it helps in nutrient uptake. It increases yield. We've tested at least 3 to 5 times, and every time it's over 30%. And when we combine it with some other products, we can almost double the yield with increased nutrition. So what we would like to do is apply these and similar products in gardens, in cities, like Poonacha was saying. Also other efforts, even agriculture and farming areas, especially when plants are being transferred from greenhouse to the field. It gives a great opportunity to give them a good start with a healthy bacteria.
So Poonacha, I think we have our homework cut out.
Yeah,
How, how do we collaborate with Britt and Rajnish and Phyllosphere in this whole program, starting with Lake Nona? But I think we have a template. We have a formula on how we can proceed with just the plants and the fruits and the vegetables and this technology in Lake Nona. And I trust that you will continue this conversation with Rajnish and Britt. Anything more you want to say? Because I want to now move to the philosophical.
I had one question, Deepak, just to continue. So, Dr. Britt, is this like FMT? Would you consider this a fecal microbial transplant? Is what you're doing with Phyllosphere analogous to FMT?
Very much so. And we've modeled many of our experiments on fecal microbiome transplants. It's both a fantastic tool for understanding. So, what part of what we don't understand about the microbiome, whether it be human or plant, is what makes it right. What makes it a good fit for that particular host. And when we, without that understanding, we're far from being able to make a universal probiotic that would work across all humans with different diets, with different backgrounds. We're getting closer in terms of human biology, and in part realizing that there's no such thing as a healthy microbiome. There's so many different types of healthy microbiomes, and we just need to understand the more global signatures of a healthy microbiome. And so the Phyllosphere product, we tend to think of it, or I tend to think of it as redressing that initial birth process. So when we're growing a plant or starting a plant in a greenhouse, it's not being exposed to natural diverse microbes, right? And we saw that as a problem that needed a solution. And this is very similar to a colleague of mine, Lindsay Hall, who works on babies that are premature and developed via C-section who've been on heavy antibiotics. She's been developing very successfully probiotics for those infants. And I see a lot of parallels here where we're redressing this problem of initial colonization, initial growth of microbes, and making sure they will, an infant or a new seedling will be colonized by something. And the point is, we don't want it to be the pathogens. We want it to have a healthy start, which is where we're coming from here. So yes, and then going back to the FMT, that is more of a something that happens later on if you have a, quote, dysbiosis. So when things go wrong with your microbiome, that's a time to attempt to purge your current microbiome and replace it with an entire community of another organism. And so, yes, we can do that at the adult plant level too, and it works. It's remarkably successful. Yeah, it's exciting, but also a very useful tool as a scientist to be able to do that.
Well, Poonacha, any more questions?
I'm good.
Okay. Rajnish, anything else you want to say? Because I want to take this slightly in another direction for the next 5 minutes before we run out of time.
I'm looking forward to that direction, and let's do it.
It's a question more for Britt than anyone else. So, Darwin, genius that he was, had no idea about gene or DNA or microRNA or any of the things you're talking about, not to mention, how genes interact with each other. Natural selection, of course, brilliant, absolutely insight. But I want to ask you now, is it time to question strict Darwinian evolution? Because what we're seeing right now is two things. One is that it looks like ecosystems evolve rather than a single gene. It's the entanglement of the genetic information, both in the soil, in the plant, elsewhere. I was fascinated by what you said about infant formula, because even if we reproduce everything about the infant formula that replicates mother's milk, there's the cooing, there's the cuddling, there's the
Yes.
kissing, there's the touching, there's the contact of the baby, which is both emotional but also microbial exchange. They're exchanging genetic information. Um, is it time to think of rethink evolution? Now, I don't want to risk anyone's reputation here because everybody here is aligned with science, and so am I. But there is some talk right there in evolutionary circles about evolution possibly being guided by consciousness, and that genes are really the symbolic representation of modes of knowing, modes of
Hmm.
experience, modes of intelligence in an ecosystem, and that we might have to revise our theories about consciousness altogether. And if we do so, we might get better technologies, including Phyllosphere, but many more, because our current understanding of evolutionary theory is based on a model that may not be 100% accurate at the moment.
Great. Yeah, I think it's a wonderful question, and I'll start by saying I always think we should be revising and rethinking and making sure. I don't think there's any use in holding on to an idea or a theory that might be changed in light of more recent data. That said, I think Darwin might, of course, there's so much he didn't know. And so when we've been revising evolutionary theory, there's been a huge amount of effort of thinking about multi-level selection, which Darwin didn't really think on that level. But this idea that you can simultaneously have selection on genes, on interactions among genes, and on species interactions, and on the community level, group selection, community level selection, ecosystem level selection. These are multi-level selection processes. And I don't think there's any doubt that selection is happening on all of those levels. You can see it happen with a selfish gene. You can see, I work in part on these bacteriophage viruses, and you can see this is a virus that integrates, that becomes part of the host genome. But if things become stressful, it jumps straight out and goes into another host genome. And so selection can act just on those genes of that virus. But that doesn't mean that it's not acting at the level of the bacteria when the bacteria has that virus gene inside. And it doesn't mean that it's not acting on the microbial community when those microbes are inside of a host together. And so one way I like to think about it is thinking about actors in a play, right? A play is an emergent property of all of the actors involved. And so when a critic comes and watches a play, they are critiquing the individual actors. So they would say, Deepak was a star in this play. He led, transformed the character in this particular way. But they might not have that same critique of you in a different play, or if it was just you and you didn't have your supporting actors, for example. And so that's a way of thinking about this multi-level selection, where you can have selection acting on the individual actors as well as on the play. And in the end, the evolutionary outcome depends on how those different selection pressures interact together. So if they're in opposition, so a pathogen, for example, selection acts on a pathogen to reproduce, but that comes at a cost to its host. It can kill the host. Those are opposing selection pressures. But often a symbiont or a member of our commensal microbiome, you could think of our fitnesses as really well aligned. So if I do well and reproduce and pass those microbes on to my children, then that's selection on both individuals in the same direction. Okay. So why am I saying all of this? I'm saying this because there's a balance in terms of how evolution acts at the whole ecosystem level that is an emergent property of how everyone is interacting together. And there's no such thing as a gene in isolation. Genes are never in isolation. Genes are always in the context of a genome. They're always in the context of a population and a community and an ecosystem. And so every time that we change something on Earth, when we build a city, when we introduce a new chemical into the soil, the reason it's so hard to predict how evolution will happen is because there are so many different levels of selection. And so, oh, sorry, yes, please, go ahead.
Go ahead, finish what you're saying.
Well, so I was just going to say that I don't actually think that there's need to throw out any of the original ideas. I think they're still very solid and they're still our guiding principles. But thinking about how they play out at these different levels and thinking about how they can be integrated into this new knowledge, I think it can only move science forward.
So then this is going to be my last question in addition to one. Would you say that with all these actors playing different roles, including some that we might perceive as villains and some as heroes, some as sacred and some as profane, but the ultimate expression of all these characters in an ecosystem is self-organization and self-regulation of the entire ecosystem. In other words, homeostasis. Would you agree to that particular point?
Yes, with one nuance.
Yes.
Which is there's often a lag. So it is hard to get to that point at this, especially when you're talking about microbes and plants and humans with such different generation times. And so oftentimes what we see in evolution is because things are happening on different timescales, sometimes, evolution overshoots, if you will. And so you end up somewhere that's suboptimal because the whole world is shifting. And I mentioned the Red Queen theory when I talked to myself.
Mm-hmm.
The thing that I really love, the Red Queen theory, in brief, comes from Lewis Carroll's Through the Looking Glass. When the Red Queen is running, when Alice is running, and the landscape is changing with her. And the Red Queen says, see, you have to run as fast as you can to stay in the same place. And this is how I think about the world, where all species on Earth are co-evolving. But the problem is that there's this optimization is happening on different timescales, in different ways, in different environments. And so I don't believe that there's ever an end. There's not an end point. And so I don't think of homeostasis as a fixed, stable point. I don't believe that there is such a thing as stability in terms of ecosystems. I think it's all very dynamic. And a lot of that dynamicism comes from selection acting on these different levels, on different timescales, on different types of fitnesses and different interaction types.
Okay, I'm going to let you go very fast, but you mentioned fungi right in the beginning. Is there something special about fungi? Because, I recently spoke to somebody in British Columbia who was talking about mother trees and fungal networks and how mother trees take care not only of their own species, but other trees as well. Are fungi very much a part of the neural network of our planet in some way?
I think the answer to that is almost certainly yes. I would say microbes more generally, bacteria as well, but certainly, okay, I will answer that question based on, my collaborator Jessica Metcalf at Princeton and we developed a very blue skies model recently that asked a question that I think is relevant to you, which is, as a host organism, as a human or a plant, what could we learn from bacteria or fungi? What do we learn from our environment? And why would we listen to these cues that bacteria or fungi give us? And it turns out that we did this big meta-analysis that all organisms on Earth use signals from bacteria and fungi, and in some cases viruses. We use signals to make hugely important decisions about our development timing, when we flower, when we grow, how much we grow, and so forth. And so I think of them in part as information. And if you're a seed arriving in a new soil, the information that's there, so much of that information is actually carried by what bacteria or fungi are there right now. And the ability of that plant to tap into that information and optimize its own growth and function in that environment is, I think, underestimated.
Last question. We are using technology right now. We're on Zoom. We now understand epigenetics. Presumably, right now, we are changing each other's activity in the brain, including gene expression. So technology has become part of the evolution of the human species, number one. And that is affecting the evolution of other species. And again, not from a scientific point of view, because I'm not qualified to answer this question, but you are. Are we changing the course of planetary evolution? And also, are there examples of evolution speeded up, like, doesn't take eons of time, happens in less than a generation?
Well, by definition, it can't happen in less than a generation. Not true evolutionary change, because that requires heritability and change in gene frequencies over time, or allele frequencies over time. But that's not to say that we as organisms are not changing within a generation. We are. And those changes, those epigenetic changes, those changes in gene expression, they can have repercussions for evolutionary time. Certainly, they can have repercussions for who reproduces, which genes are passed on, and so forth. To your question of whether we can speed up evolution, though, I mean, the answer to that is very much yes. And I have no doubt that we are doing that at the moment in a absolutely unprecedented way with climate change. We're seeing very dramatic effects on populations of plants and animals in response to this incredibly rapid changing climate, changing species interactions, increased pathogens as we move organisms around the earth. We've sped up the rate that pathogens are spreading, as we unfortunately are seeing right now in a very large way with this pandemic. And so all of that, well, let me say this way. The speed of evolution is driven by two things, and only two things as far as I'm concerned: the strength of selection and how much variation there is. And that variation is, how large the population sizes are, how much mutation there is, and so forth. But basically, if you have large populations and a lot of genetic variation, then the stronger selection is, the faster evolution happens. And what we are doing right now is absolutely ramping up selection pressures on Earth in a way that we've never experienced before. And so, yes, we are speeding evolution up.
This has been an amazing conversation. Very grateful to you and your insights and your expertise.